Hello, my name is Adrian Newman Tancredi, and I'll be talking to you today about cortical 5-HT1A receptor biased agonism as a novel mechanism for therapy of treatment-resistant depression. Now, depression is a major medical need. There are many millions of people affected, and the yearly prevalence is around 5 to 10%. And this has a major economic impact and social impact. There is therefore a need for novel antidepressants because the existing ones are not as effective as we would like. In fact, there has been little progress for most of the compounds since amitriptyline was identified about half a century ago. So there's a need for novel, improved antidepressant drugs. And ketamine has given people a lot of hope and interest as a novel treatment. This publication from 2006 by Campbell Zarate and colleagues caused much excitement. It's been cited over 3,000 times, and it showed that a single administration of ketamine was sufficient to produce very rapid antidepressant effect within a matter of a couple of hours. And these effects lasted days, even up to a week, and therefore it was hailed as a step forward in the treatment of depression, and particularly of treatment-resistant depression, and even hailed as a cure, potentially, for depression. Of course, the reality is that ketamine is not an ideal solution. As well as these striking antidepressant effects, it also has a number of side effects which limit its use to medical supervision, and the long-term use of ketamine is still under discussion in terms of its safety and efficacy. So overall, it's been a step forward, but it has significant side effects. But the discovery of ketamine's antidepressant properties has stimulated a lot of interest in the underlying mechanisms of action. Because, of course, it acts as a non-competitive NMDA receptor antagonist, which has effects on the glutamate system. But ketamine also has other effects, and notably on the serotonergic system, which lead to activation of serotonin receptors, and particularly 5-HT1A receptors. Indeed, ketamine has been known for a long time to inhibit 5-HT uptake, and as well as synthesis and metabolism in a series of different tests. And the depletion of serotonin neurotransmitter stocks in the brain prevents the antidepressant effects of ketamine. So it is necessary to have serotonin present for ketamine to be active as an antidepressant. And other studies have shown that the increased levels of glutamate, which are triggered by ketamine, activate AMPA receptors. And this is important for ketamine's antidepressant activity, but AMPA receptor activation itself also increases serotonin levels in a particular brain region, which is the prefrontal cortex. So once again, there's an involvement of serotonergic function in the effects of ketamine. And finally, if a particular subtype of receptor, which is the 5-HT1A receptor, is blocked by 5-HT1A receptor antagonists, then ketamine's antidepressant-like properties in rodent tests are blocked as well. So this demonstrates that ketamine's antidepressant effects are mediated by a number of different systems, glutamatergic, but also serotonergic, and that, at least in these rodent tests, they necessitate activation of 5-HT1A receptors, particularly in the frontal cortex. So it's important to understand that Serotonin receptors, 5-HT1A receptors, are expressed in a number of different brain regions, including the raphe, including brainstem, hippocampus, and the frontal cortex. And depending on which brain region these receptors are expressed in, they will signal through a number of different systems, different G proteins, different intracellular mechanisms. And in the cortex, the ones which are particularly important are ERK phosphorylation, BDNF release, and mTOR. And all of these have been strongly associated with antidepressant properties. Indeed, the cortical 5-HT1A receptors are the ones which are very strongly associated with the activity of ketamine and other antidepressants, whereas 5-HT1A receptors in other regions mediate other responses. And in fact, 5-HT1A receptors in the raphe have been shown to actually slow down the activity of antidepressants which function by reuptake inhibition. It is therefore important to target 5-HT1A receptors in the cortex whilst avoiding activation of 5-HT1A receptors in other regions, and particularly in the raphe. Now, to do that, you would therefore like to have an agonist, an activator, which would be biased or preferentially activate 5-HT1A receptors in the cortex without influencing the activity in other brain regions. So that's been the work that we've been carrying out and looking for such biased agonists. And we've identified a compound which we call NLX-101. It's also known as F15599. And this is a first-in-class cortical 5-HT1A receptor biased agonist. In an early study a few years ago, we showed that NLX-101 stimulated energy consumption preferentially in the cortex using an fMRI, magnetic resonance imaging study. And you can see that here on this image, particularly in the cingulate cortex, you see very selectively activated in that brain region, whereas another agonist, F13714 or 8-hydroxy-DPAT, didn't have such precise preferential biased agonism in the cortical regions. And there's an antagonist, MPPF, which didn't activate at all. So this was a brain imaging study which showed this biased agonist property of NLX-101. So, in fact, NLX-101 is an exceptionally selective ligand for 5-HT1A receptors. In this receptor profiling study that we did, we showed that NLX-101 has high affinity for 5-HT1A receptors. That's in the bar on the left there, whereas it has negligible activity at a whole range of other receptors. And this distinguishes the compound from other serotonergic compounds such as buspirone or tandosperone or flibanserin, which are not selective like NLX-101. Also, NLX-101 has this preferential intracellular effect to activate ERK phosphorylation, as opposed to other signaling cascades. And as we just mentioned, ERK phosphorylation is important because it's associated with antidepressant effects. So this activity, we think, is interesting in the context of discovery of novel antidepressants. And in fact, when you go to rodent tests of antidepressant-like activity, we see that NLX-101 has very striking effects. Now, we use the RAAD forced swim test. This is a classic test of antidepressant-like activity. And what you see here is that NLX-101 could completely reverse the immobility behavior, which is a marker of depression-type behavior in this forced swim test. So that says that the compound has robust antidepressant-like activity, even on a single administration. And that's something that you can't achieve with reuptake inhibitors such as citalopram, which require repeated administration. You can do it with ketamine, but at much higher doses. Now, and the second thing to notice from this slide here is that the effects of NLX-101 are sustained over several hours. And you can see that even after 8 hours, NLX-101 still completely reverses this immobility behavior in the forced swim test. So that's a very robust response. And we also tried putting NLX-101 directly into the cortex of the rats by microinjection. And once again, we found that through this route of administration, we could reverse the immobility behavior. So that tells us that it is the cortical 5-HT1A receptors which are mediating the antidepressant effects of the compound. We also looked at another antidepressant model, which is the chronic mild stress model. This is a robust test of antidepressant type effects. It consists of subjecting, in this case it was rats, to a series of mild stresses. And after a certain number of weeks, the rats will become anhedonic, which means they will reduce their consumption of a sucrose solution, which they would normally drink more of. And this is a marker of depression type behavior. That can be reversed, as in these studies from Marius Papp's laboratory in Cracow. It can be reversed by treatment with a classic antidepressant, which is imipramine, and it takes about 3 weeks to re-establish normal sugar drinking behavior. Ketamine is much more rapid. Even after just 3 days under similar conditions, you see a response. But what's interesting is that in the case of NLX-101, a complete rescue of the anhedonia behavior was seen even just after a single day. That means that it's more rapid than the other antidepressants which have been previously tested. And this was really very striking. A second striking result from that study was that even after administration of NLX-101 was stopped, the anhedonia behavior remained rescued. In other words, the animals continued consuming the sucrose solution even without the continued administration of the drug. So that looked like there was some modification in their neuronal structure and in their responses, which was a lasting modification even after discontinuation of treatment. So the question is, well, what is actually going on here in terms of the neuronal responses? And we think that there's a convergent mechanism of the activation of cortical 5-HT1A receptors, which converges with that induced by ketamine. And it's something like this. So firstly, both ketamine and NLX-101, the 5-HT1A biased agonist, target, we think, the same neurons in the frontal cortex, and these are the GABAergic interneurons. And both activation of 5-HT1A receptors and blockade of NMDA receptors produce a reduction in GABAergic activity. And this has the effect of reducing GABA release, which therefore means that there's less GABA-A receptor activity on the glutamatergic pyramidal neurons. A reduction of GABA-A activity means less inhibition of the neurons, and therefore an increased amount of glutamate release, which would therefore stimulate postsynaptic AMPA receptors. And that activation of AMPA receptors triggers the cascade, which includes AKT, mTOR, ERK phosphorylation, BDNF release, as well as plasticity and neurogenesis effects, which are longer lasting and therefore provide the sustained antidepressant effects of these compounds. And to support the conclusion that this is what's happening with this 5-HT1A biased agonist, we looked at a number of different models, including the effects of NLX-101 on pyramidal neurons in the frontal cortex. And yes, the compound does increase the neuronal firing of the pyramidal neurons. Yes, NLX-101 does increase frontal cortex ERK phosphorylation ex vivo. And yes, it does increase mTOR phosphorylation in that same brain regions in the frontal cortex. And it also increases BDNF levels and neurogenesis in the brain. So all of these effects are consistent with a rapid-acting antidepressant activity similar to that of ketamine. We also showed that there was changes in dendrite length with NLX-101 in the cortex and in the hippocampus, and this was a model following a ischemia challenge in mouse. So once again, NLX-101 is producing neuroplasticity, neuromodulatory effects, which are consistent with an antidepressant effect in these different tests. And I'd just like to mention as well that in mouse, we also saw a reduction in immobility behavior in the forced swim test following single administration. And we've also seen, again, in mouse, increased ERK phosphorylation and a reversal of a sucrose consumption deficit following chronic mild stress. So this is transposable across both mouse and rat species. And what's more, given that NLX-101 targets cortical 5-HT1A receptors, this suggests that it might have additional effects, not just on mood, but also on cognitive properties. And that's exactly what we see. In fact, chronic mild stress produces a deficit in certain cognitive functions, notably in novel object recognition tests, and that is completely rescued by treatment with NLX-101. And we also see that NLX-101 is active in a whole battery of other tests on memory and cognition in rat models. So taken together, what this says is that targeting cortical 5-HT1A receptors with a biased agonist which avoids activating 5-HT1A receptors in other brain regions, this seems to be a promising strategy for improved therapy of depression, at least based on a series of animal tests. And based on these results, we've launched a campaign of drug discovery, and we've identified novel antidepressant drug candidates based on NLX-101 and similar chemical structures, but potentially having even further accentuated antidepressant effects and further accentuated bias for particular receptor subpopulations, and notably in the frontal cortex through activation of ERK phosphorylation. So overall, what we've seen in this presentation is that depression is a major healthcare challenge, and that ketamine, although it's a useful addition to the pharmacopoeia for treatment of depression, is not a perfect rapid-acting antidepressant. And in fact, a step forward would be maybe to directly target 5-HT1A receptors as antidepressant mechanism in the frontal cortex. And NLX-101 is in fact a selective cortical biased agonist. It has effects on all these markers, ERK phosphorylation, mTOR, BDNF, neurogenesis. It shows robust antidepressant activity in the mouse and rat, has pro-cognitive activity, and has really provided a very interesting prototype for discovery of novel biased agonist antidepressant drug candidates. Thank you very much for your attention.